स्पर्मेटोजेनेसिस। आज हम स्पर्मेटोजेनेसिस के बारे में क्विक रिवीजन करेंगे। तो स्पर्मेटोजेनेसिस इज अ पार्ट ऑफ गैमेटोजेनेसिस। इट इज अ पार्ट ऑफ गैमेटोजेनेसिस। गैमेटोजेनेसिस is of two types, spermatogenesis and oogenesis. It occurs in male and oogenesis occurs in female. The location of spermatogenesis is seminiferous tubule of testes, seminiferous tubule of testes, and in females, cortex of ovary. So gametogenesis is the process of formation of gametes, and it takes place in gonads. The seminiferous tubule of testis and cortex of ovary. So in this lecture, we will concentrate on spermatogenesis. So here is a slide about gametogenesis. Definition, you can see gametogenesis can be defined as the process of conversion of PGC, primordial germ cell, and their maturation into male and female gametes. In males, it is called spermatogenesis, and it takes place in seminiferous tubule of testis, and in females, it is called oogenesis and takes place in the cortex of ovary. These are the few events in the gametogenesis. First is the formation and migration of PGC and their differentiation into male and female sex cells. Second is the mitotic division. Third is the meiotic reduction of DNA or chromosome content. And fourth is the differentiation. Out of these four events, the first two events takes place in both spermatogenesis and oogenesis. But from the third and fourth point, this third and fourth point is different in both spermatogenesis and oogenesis. So moving further, if we look at the time period, we can define the spermatogenesis into two parts. The prenatal part. Natal here is the birth, so before birth, and the postnatal part, the after birth. So we look at the time period. This is the prenatal duration and the developmental events and details. During the second week, you can see the formation of primordial germ cell in epiblast. Epiblast is the, and there is a layer hypoblast. So from the epiblast, the formation of PGC begin. During the fourth week, migration of PGC to yolk sac wall. During the fifth week, PGC reach the developing gonad. During sixth week, gonadal differentiation occur, and then PGC proliferation, that is mitosis, and then after this, influence of SRY gene on Y chromosome. So these are the events, the prenatal events, which takes place during the formation of spermatozoa, or we can say spermatogenesis. This is the picture of male reproductive system, adult male reproductive system. You can see the urinary bladder. This is the rectal part, seminal vesicle, urethra, and epididymis, and this part is the testis, where the process of spermatogenesis. If we look closely to the testes, we can see the layers, tunica vaginalis, albuginea. These are the covering of testes, and this is the seminiferous tubule. This part is the seminiferous tubule, where the process of formation of spermatozoa takes place. And moving further, as you can see, this is a tube-like structure. Further, its storage and transfer occur through whole of this system. So you have to remember the seminiferous tubule of testes. This is the another picture, cut section of seminiferous tubule. You can see the different cells are present. Spermatogonium, you can see the primary spermatocyte, you can see the secondary spermatocyte, spermatid, Sertoli cells are the supportive cells. And it also provides the nutrition. And you can see that there are different shape and size cells are present under the cut section of seminiferous tubule. This picture presenting the migration from the epiblast This portion is the epiblast towards the yolk sac. Now we come to the spermatogenesis. It is a process of maturation of male gamete in a wall of seminiferous tubule. It involves a series of changes leading to the conversion of spermatogonia to the spermatozoa. It means the time period for this is the formation of gamete takes place only during the reproductive period. This begin in the prenatal life, then again stop at the time of birth, and it resumes the process in the reproductive life under the influence of hormone, or we can say the age of puberty, that is 12 to 16 year for male, and continue even into the old age. The duration for the whole cycle, whole of the process of spermatogenesis is around 64 to 74 days. From this flow chart, you can see the prenatal part, then the birth, and after puberty, and this part is showing the about the reproductive stage. So at prenatal part, primordial germ cell or yolk sac multiply to form the spermatogonia, and spermatogonia cell undergoes the resting phase, and at the time of puberty, that is 12 to 16 years, it resumes the cycle again. Spermatogonia mitosis division occur, and it divided into spermatogonia type A, which is little bit dark in color, and spermatogonia B, which is pale in color. It's not dark, it's pale in color. So spermatogonia convert into the spermatogonia type V. The spermatogonia A act as a reservoir cell, cells, and spermatogonia B participate in the further process. It undergoes the mitotic division, and it gives rise to the primary spermatocytes, and then this process of conversion of spermatogonia to primary spermatocyte, this is the first stage of the spermatogenesis, that is spermatocytosis. It takes around 16 days. Duration is around, it takes 16 days around. The second stage is the meiosis. The meiosis occurs in two parts. The first meiotic division give rise to the secondary spermatocyte, and there is a reduction of chromosome, and in the second meiotic division it gives rise to the spermatid. So up to this stage it is meiosis, in meiosis 1 and meiosis 2. Meiosis 1, there is a reduction of chromosome, but in meiosis 2 there is no reduction of chromosome, and it is also called equation meiosis. And after this, the spermatid metamorphosis occur and convert into the spermatozoa. So this stage is called, the third stage is called spermiogenesis. Out of this, spermatocytosis takes around 16 days, meiosis takes around 24 days, and spermiogenesis also takes around 24 days. This is flow chart. You can see spermatogonia, and it divide after the mitosis division, it divided into dark type A and the light type B. It further go under the process and give rise to the primary spermatocyte. Then meiotic division 1 and meiotic division 2. So this process comes under the spermatocytosis. And this comes under the meiosis. Takes around 16 days. Takes around 24 days. And as I told you earlier, the meiosis 1, the chromosome number become half. Here it is 44 plus XY, here it is 22 plus X. But during the meiotic 2 division, you can see there is no reduction in chromosome. That's why it is also called equation meiosis. And after this, the metamorphosis, the third stage, that is spermiogenesis process. It also takes around 24 days. And you can see rounded spermatid convert into a motile structure, that is a spermatozoa. So we will look into the detail the process of spermiogenesis. So this is a picture representing the process of spermiogenesis. So firstly, what is spermiogenesis? It is a process of metamorphosis of a rounded spermatid to a spermatozoa, that is motile structure. You can see both pictures are completely different, and this takes place through the metamorphosis. Secondly, it takes around 24 days, and this is the third stage of spermatogenesis. Okay, so you can see here, spermatids is more or less a circular structure. It has a cell membrane, nucleus, Golgi apparatus, mitochondria, and the proximal centriole and the distal centriole. All these structure metamorphize and give rise to the spermatozoa. So, what are the major events which occur during this process? First is the nuclear morphogenesis, or we can say condensation. Second is the Golgi apparatus. Golgi apparatus becomes the acrosomic cap, and it covers around the 2/3 of the head. And mitochondria surrounded by this middle piece. And the centrioles, the proximal centriole, you can see firstly they are close to each other, lies in the neck part. Proximal centriole and distal centriole lies at the end of the middle piece. And axial filament arise from these centrioles and enlarge and grow to form the tail piece. So now we got the we got this spermatozoa, head part, neck part, middle piece, and the principal piece. The first, there is a nuclear condensation because head have to bear the most insult during fertilization, uh, the pressure effect. So it condensed to bear that effect. Second, the Golgi apparatus, or we also called it acrosome cap. It secretes the enzyme lysozyme, and which secrete the hyaluronic acid, which help in digest the ova membrane during fertilization. Next is this proximal centriole. Its function is to provide this axial filament, which is the main element for the movement. Next is the mitochondria. For the movement, it requires energy, so mitochondria help in generate that energy. So in this picture, you can see clearly there is a change in shape, and this whole process is called metamorphosis. The nucleus, the arrangement of structure occur, the condensation of nucleus occur, then there is a formation of this tail part. You can see here, this is the formation of tail part. And the centrosome, firstly you can see close to each other, and later on proximal centriole, and here it is distal centriole, and rearrangement of all the organelle occur. And finally, there is a shedding of excess cytoplasm, and that excess cytoplasm, where it goes, it is engulfed by the Sertoli cell or the supportive cells. Next, in middle piece, you can see this proximal centriole, and this annulus part is surrounded by the mitochondria. It is the primitive form. This together is known as middle piece. And what happened with the cell membrane? Cell membrane persists, and it helps in spermate recognition, spermate recognition, spermate binding, and spermate fusion during the fertilization process. So here it is a quick review about spermatogenesis. It is a part of gametogenesis. You have to remember its definition, its location, then the flow chart, the prenatal changes, and the what is the resting phase, and then the postnatal changes, and after this the important phase, that is the spermiogenesis.